Gene therapy and applications. Novel lentiviral vectors for gene therapy of sickle cell disease, combining gene addition and gene silencing strategies. By Sky, Charlotte, Annetta, and Caitlin.
Sickle cell anemia is a genetic disease that affects the hemoglobin protein and, in turn, the shape of red blood cells in the body. 20 million people worldwide are currently affected by the disease, although it is more common in certain ethnic groups, including African Americans, Hispanic Americans, and people of Middle Eastern, Asian, Indian, and Mediterranean descent. Sickle cell anemia can lead to serious health complications, including stroke, fatigue, infections, pain, and eye problems. A mutation at the genetic level, such as the GAG to GTG mutation at codon beta 6, carries through to the hemoglobin protein being disfigured, which is what we call a sickle cell. In order for a person to have sickle cell anemia, they have to have mutations in both copies of their beta globin gene, also known as HBB. Sickle cell anemia was first discovered in 1910, and effective cures and treatments are still being investigated. As the article discusses, a treatment involving the transportation of stem cells in patients was thought to be effective. However, after a trial, the sickle cell count was still elevated in patients. Other treatments involving gene addition and gene silencing separately have also been tested, although researchers are still searching for a more effective treatment. The purpose of this article is to evaluate the efficiency of lentiviral vectors for gene therapy of sickle cell disease. The therapy, which combines gene addition and gene silencing strategies, aims to lower HBS levels, which is the sickle cell allele, and ultimately correct the sickle cell phenotype. The article follows the development of lentiviral vectors used to insert a variant form of the beta globin gene into the hemoglobin units. The aim was to use the developed lentiviral vectors to downregulate either the BCL11A gene or the beta S globin gene, which was projected to increase the levels of therapeutic beta-like globin levels without increasing mutations, and in turn correct the beta hemoglobinopathy phenotype.
In order to deliver the therapeutic gene modifications, lentiviral vectors, also known as LVs, are used as carriers due to their ability to transfer and integrate a transgene of interest into target cells. The transgene of interest is incorporated into the lentiviral vector DNA, which then transduces the target cell genome and initiates its effect. In the case of sickle cell disease gene therapy, it is the downregulation of the BCL11A or BS globin genes. The LV used, which was carrying an anti-sickling beta-globin transgene, was under control of the HBB promoter and a mini-locus control region. The study focused on the use of two bifunctional lentivectors combining a BAS3 gene addition approach with an AMI-RNA-based gene silencing strategy. These LVs were inserted into hematopoietic stem and progenitor cells, HSPCs, which are healthy stem blood cells from sickle cell disease affected individuals that can create new blood components or self-renew. The first of the two bifunctional lentiviral vectors combines the BAS3 gene addition strategy with the insertion of the MIR BCL11A into the second 256 nucleotide long intron position of the BAS3 transgene. This addition of the AMI RNA and the MIR BCL11A consequently results in the silencing of the BCL11A XL that encodes for a zinc finger protein which represses the Y globin expression and HbF expression in erythrocytes. The second of the two bifunctional lentivectors combines the BAS3 gene addition strategy with the insertion of the MIR7M, as it was the most efficient miRNA. In order to generate an effective miRNA that would specifically target the beta-globin gene, a sequence of small interfering RNAs and shRNAs targeting HBB were inserted into the backbone of MIR223 and modified via the removal of 4 nucleotides at the 5' end and addition of GC-GC segment at the 3' end. The RNA effectiveness was determined through the transduction of the K562 cells with the LVs containing the miRNA of interest in order to assess their silencing effect on the BAS3 gene. The bifunctional lentivector was then utilized to transduce primary adult HSPCs from sickle cell disease donors. The BAS3 MIR7M lentiviral vector was of particular interest in sickle cell disease gene therapy due to its effectiveness in the downregulation of the BS globin gene, therefore making it a promising avenue for the incorporation of BAS3 in HB tetramers.
These lentiviral vectors essentially both added a therapeutic gene while silencing one that was problematic. This combined gene addition and gene silencing approach aimed to improve gene therapy outcomes for sickle cell disease and beta thalassemia. The improved treatment for beta thalassemia through the combination of the therapeutic beta AS3 gene and the downregulation of BCL11AXL reactivated gamma globin. Ultimately, this improves the phenotype for beta thalassemia. This result suggests a potential shift from merely managing symptoms to addressing the root cause of the disorder. Within this particular study, the researchers found that the new vectors, beta AS3M/MIR7M, did not show any adverse impacts on HSPC cells in regard to their viability or differentiation capacity. Furthermore, it effectively reduced the expression of problematic HBS without affecting their beneficial beta AS3 expression. In comparison with other therapeutic strategies, this paper discusses the advantages of using AMI RNA over other silencing techniques like shRNA. The latter can be toxic, interfere with natural RNA pathways, and trigger immune responses. In contrast, AMI RNAs, especially in the lentiviral approach, present fewer risks. Although genome editing can draw some concerns, the researchers have demonstrated that the lentiviral vectors is a promising and safe strategy that may offer an alternative or complementary approach to genome editing. Therefore, the overarching implication of the article is that the described lentiviral approach could shift the objective of gene therapy for sickle cell disease and beta thalassemia. Instead of just reducing symptoms or the need for blood transfusions, the therapy could aim for a complete cure of the disease.
The research on lentiviral vectors for gene therapy in sickle cell disease highlights the immense promise of gene editing technology, but this potential is accompanied by intricate ethical considerations that require careful scrutiny. The study's goal to correct the sickle cell phenotype through gene addition and gene silencing strategies underscores the importance of understanding and addressing these concerns within the context of the research. A central ethical concern is the precision and safety of gene editing using lentiviral vectors. While lentiviral vectors offer targeted gene modification, the possibility of off-target effects could lead to unintended consequences. Ensuring the accuracy of these vectors is vital to prevent unintended genetic alterations. Addressing these concerns involves refining the vector's specificity through vigorous research and validation. The heritable nature of gene editing poses another ethical issue, which is particularly relevant to lentiviral vector-based therapies. Modifying patients' genetic material using lentiviral vectors could result in permanent changes that are passed on to future generations. This raises questions about the broader societal implications and the morality of permissibility of altering the germline. Deliberations on the long-term consequences of heritable gene modifications, especially in the context of lentiviral vectors, are crucial considering both potential benefits and unforeseen risks. Equity in access is a pressing concern with direct relevance to lentiviral vector technology. The transformative potential of gene editing using lentiviral vectors must be harnessed to benefit all, irrespective of a socio-economic status. Collaborative efforts are essential to ensure that these therapies are affordable and accessible, averting the exacerbation of existing health inequalities. The dual-use dilemma, while not exclusive to lentiviral vectors, adds another layer of complexity to the ethical landscape. While intended for therapeutic purposes in the context of lentiviral vector-based gene therapy, gene editing could be misused for non-medical enhancements. Stringent ethical guidelines and regulatory oversight are vital to strike a balance between legitimate therapeutic applications and potential misuse of lentiviral vector technology. Long-term safety monitoring, including assessing genotoxicity, is paramount, especially when utilizing lentiviral vectors. Given the novelty of gene editing using lentiviral vectors, post-treatment surveillance is necessary to identify any delayed adverse effects that may arise over time. Rigorous monitoring protocols should be established, particularly when lentiviral vectors are employed, to ensure patient safety and well-being. To comprehensively address these concerns in the context of lentiviral vector-based gene editing, researchers must prioritize enhancing the precision of lentiviral vectors to mitigate off-target effects and genotoxicity risks. Ethical governance specific to lentiviral vector technology should be reinforced, incorporating rigorous review processes and adherences to established guidelines. Public engagement emphasizing the role of lentiviral vectors ensures that diverse perspectives are considered, fostering a more inclusive approach to ethical decision-making. Furthermore, continuous dialogue among stakeholders, including researchers, ethicists, patients, and regulatory bodies, is essential to navigate the evolving landscape of gene editing using lentiviral vectors. Striking a harmonious balance between scientific progress enabled by lentiviral vector technology and ethical responsibility is paramount for successful development and deployment of gene editing technologies. In summary, the researchers' exploration of lentiviral vectors for sickle cell gene therapy unveils a powerful tool with vast potential. However, the associated ethical concerns spanning accuracy, heritability, informed consent, access, dual use, and safety must be acknowledged and resolved within the research context. By robustly addressing these ethical considerations, particularly in the specific context of lentiviral vector gene editing technology, researchers can pave the way for responsible and beneficial gene editing applications that align with both scientific progress and human values.